Hi, I'm Charlotte. I am a senior software solution engineer with Neo. I've been around here for like 8 years or so, and I work with Alex and help other people do their sort of pharma stuff. Let me share my screen. And the good thing is you get to, so far we've watched everyone having lovely stuff with slides and things, and now you get to watch me mess up with a demo because that's the way the demo gods work, which is lovely stuff. So hopefully we can see my screen. Yes, lovely. Okay, so we're going to talk about, I'm going to talk about GenAI knowledge graphs, basically giving your LLM a left-handed brain. So let's start with a little bit of a definition. So far, everything we've talked about so far has been quite abstract. We've talked about what graphs are, but we've never really discussed what goes into making a graph. So in a scientific world, we're looking here, for example, we're looking at some sort of documents and things, and data starts with things. So we have documents, we have references, so documents reference things, we've got chunks, which are parts of documents, and we have named entities from those documents. Those named entities might be things like molecules, diseases, lots of other varieties of different things, right? So that's what we have. This is data. This is what we have in a scientific world. And we want to take those and turn them into, in our graph world, nodes or entities. Things have properties. No big surprises there. We have a variety of different properties. Properties within Neo4j's context are key value pairs. So a reference might have an ID. All of these have IDs because that's the way we happen to roll. Documents can have names, titles. A named entity might have a name, so that might be the molecule name. And chunks are texts and vectors so far for doing all your standard sort of GenAI stuff. So far, so everyday database. But the thing which obviously brings a graph database into the fore is when we add in relationships. So those things are all related. A document cites a reference. A document mentions chunks, so a document is made up of chunks. A document also mentions named entities, and chunks have named entities as well. So your molecule will be part of a chunk, which is part of a document, and that document might cite something else. Right, so now we have ourselves a knowledge graph. And what we have basically, for all intents and purposes, is we have a way of representing real-world data in an abstract form. Right? We don't actually have named entity as a separate thing. It's part of a document. Okay, hopefully we're all clear on this. I can't see any questions and answers, so if there are questions and answers, just throw them out there, and I'm sure that Alex will let me know. If there's anything else, I don't think you can talk, but just go for it anyway. So how do we do this? We can have a look back at the Danish pharmaceutical use case because we're back into the scientific intelligence world. So the problem sort of comes down to it, and for full reference, I'm a software engineer, so I tend to see things as user stories. And user stories are always a principle of, as a something, I want to do something. That's the way I process problems. So my problem is that as a clinical researcher, I kind of want to see what other companies have in their pipelines, partly because I want to know what else is being studied, what else is being looked into, but I also want to see if there's a trend. Maybe there's a certain set of diseases which are turning up more commonly. Maybe that's something I want to research as well. The current process for that is I go to all of the companies, I get all their pipeline documents, and then I read all those documents. There are a lot of documents. None of those documents are in the same format. I then take those documents, take all the information I got from it. Maybe I put it into a spreadsheet because I'm old school, or maybe I put it into something new, snazzy, like a notebook, who knows. But then basically I'm going to go through and I'm going to analyze, and analyze. That is slow. All of this process takes time to do it. And we know we don't want to do that. We've had OCR and things like that for a long time to try and avoid that. So let's try and avoid that again. We're going to look at it from the point of view at the moment. We're just going to look at Boehringer's pipeline from October 2024. This is what it looks like. I have the file on my laptop, so I can show you what it looks like as well in a minute or two. But in essence, it's a set of pages that define the different phases where their research is based, what they're looking into, what those are targeting, and indications, I guess, of what they're doing. We're going to use something called the LLM Graph Builder, which is a very straightforward tool, which Neo4j has produced, and you can access it right now. You could go and use it, and we'll show you that when we actually get to the sort of the live. This is definitely going to go wrong part of the demo, which you'll be excited to see. But we'll basically put the file into there. We're passing it straight to OpenAI AI. So in this case, it's going to GPT-4o, and it's going to parse that document. And we haven't told it to do anything. We've literally just thrown it in there, let it have its way. And in this case, it's brought out a set of diseases and molecules. Here we've got like a big overview of all of those diseases and molecules. We can zoom in a bit, and we can zoom in a bit more and see that, if we look at here, we can see that we've got oncology is the disease, and we've got a set of molecules which treat that disease. So they're basically aiming to try and make that better. And we have other things where we can start from the point of view of the molecules. So we can see that this specific molecule treats 4 different diseases. I'm not going to embarrass myself or all of you by having to listen to me try to pronounce those. I just thought I could try that one, but no, I've changed my mind. Okay, so with the LLM Builder, we also have a chatbot down the side. And I appreciate this is small text, but in essence, we've asked the question, what molecules or drugs are being researched to treat lung cancer? And we've only answered it for this one document, right? And it's gone through and it's found the answers and it's pulled it out and it's used the knowledge graph to answer those questions. Okay. What about the top 10 pharma companies, right? We could absolutely write something. As a software engineer, in my head, I know that I could write something that could parse the structure of the Boehringer pipeline documents, because they're probably going to be fairly consistent. I can go back and check them and I can see what I'm looking for. However, the pharma companies, the top 10 of them, are not producing those pipeline documents in ways which are particularly useful. We have the Bristol Myers one there. I think that's a Pfizer one, and a Novo Nordisk one. All of them are in different structures. Some are Excel files, some are PDFs, some are tables, some are snazzy. We've had a graphic designer come and make this look better for us style. All of this stuff is there, okay? And we need to be able to parse them all in a very easy, quick way. So in this scenario, and don't worry, we will be looking at this in proper so you get to see it. We've just added in all of those documents. We've allowed it to run through. We've asked the same question. The difference is that now we're starting to get that they're coming from different companies. So AbbVie and Pfizer, we're starting to get results from all of the documents as opposed to just the one which we put in there. And our graph has all this information in there. So let's first, before we look at how we can improve this, let's go to, I think we want to go to Chrome. Okay, right. So we have the situation where we've got one file uploaded, and I can click on this and I can preview what that graph looks like. I'm not going to do that live because I think we're all aware doing virtual stuff that the processing power that rendering this video to you is using has basically made my laptop useless for doing anything other than showing this video. So this is all pre-rendered stuff, but this is all live information we're looking at. So this is everything that was extracted from that one document. Well, most of the stuff. I can turn off all the documents and chunks, and now I'm just looking at the named entities which were pulled out. So I can see I've got diseases and molecules, and if I look and zoom in a bit further, I can see I have progressive, pulmonary fibrosis. Lucky I picked one I can read. And that's obviously a disease because we can see the node type there. We can see there's a target, which is what this molecule is targeting. So nerandomilast, is targeting PDE4B, which treats progressive pulmonary, and the other two as well. Okay, and this is what we're extracting from our graph, from the documents. We can use that. I've asked a question here: what molecules are being researched to treat lung cancer? And again, same token, because time and how long it takes to actually type these things out, I've got a response back from it. And this response is only based on this one graph. I can actually go to Aura, which is where I've uploaded this, and I can see all of the actual graph entirely. I can just run queries against it, and we'll have a look at the other ways we can access stuff later on as well. But let's have a quick look at the same set of data, just with a different. So I've now uploaded all of the documents. So I put all 15 documents into there. And I can do the same thing. I can click on my Boehringer one. I can see if I look at the preview of that graph. It's very similar, slightly different color scheme, but I can still see I've still got more stuff in here because I've pulled from a lot more different files. This has given me a whole lot of different types of entities. And you can see because we just passed stuff in, it's just dumped a load of stuff in there. So we have development with a phase of a space, development phase without the space, development stage with a space. You know, a whole load of different areas which make it more complicated for it to ask questions because we expect the LLM to be able to cope with this and to be able to understand all the different types which are through there. So I've asked the same question, you know, what treats lung cancer? And in this one here, instead of getting just the so if we go back to the Chrome one here, all of these molecules here are from the Boehringer one, because we can see they all begin with the BI bit at the beginning. And if we have a look at the one where he brought in everything else, we've started for that one's from Ave. We know we have Pfizer ones in there. So we know we're definitely pulling from all the different files. And we can preview, I don't know, those four together and see what that graph looks like. As soon as it gets there, it feels ready. Okay, maybe we're already previewing this one right now. We will come back to that. Let it do its thing in the background whilst we quickly go back to the presentation part of this. So at the moment, what we've done is we've just dumped the files in, and that's had mixed results. Okay, we can ask questions to our LLM. We can get a set of stuff out of it. In some respects, it's really good, right? We can just throw in documents and we can just read them and we can start to. We don't have to go through and do lots of analysis. But the downside is that it's a really scattergun approach. The models which are generated, one of them had like 10 nodes, one of them's got 106 types of nodes. Like, it's not practical. And as we keep adding more and more documents, it will add more and more node types to the point where it becomes almost unmanageable to deal with those sort of things. So with the LLM Graph Builder, we can add some things to improve it. And the reason why I'm going to show you this, firstly, I think it's important to notice that, well, not notice, to note that the LLM Graph Builder is a demo tool, right? This is not what you would use for production. There are lots of reasons why you wouldn't use it for production. You're passing it to an LLM that is not trained on, say, in this case, the use case data is not trained on pipeline documents, on medical data, on bioinformatics. It doesn't know particularly. It has the same problem that Alex was talking about, that you might be bringing in Active Directory versus the other ones which begin with AD. But we have all of those things in there, and we don't know because the LLM is not really capable of figuring that out straight from just passing in a document. But what we can do is we can improve this by passing in schemas. Now, myself and Alex sat down for a little while and sort of came up with what would we actually do to schema? What would this look like if we were doing a pipeline schema? So we come up with this. It's really simple. It removes the vast majority of the node types because we only actually want 5 nodes, right? We want to have the molecule which treats a disease, that molecule targets a particular target, and we hopefully get an association between them. So we know that target is associated with that disease. We know the molecule is part of a clinical trial, and we know the company has that molecule in the pipeline. So let's have and we can also, we'll carry on a little bit further because I can show you more. The other thing we could also do with the schema, or after we've done this to improve it, is we can actually improve the prompting. So what we've also added is this prompt here. You're an expert in extracting structured information from clinical pipeline documents. Identify key details such as the company name, the molecule name, what indications it treats, what it targets, and the phase of development they're at. And that every molecule will be in a pipeline for a company. All right, we're trying to tell the LLM this is the information you need in order to be able to extract this information from the document. Within the builder itself, and we are going to go to you. So I've read in the same documents as before, but the difference with this one is that in the graph enhancements, I've added in my patterns. So I'm able to define my schemas. I can type in different things here. I can add different nodes if I want to. They could be it would be very odd, but I could have a Charlotte node, and I can just create that. I can say it's going to be particularly with association, add my own relationships. What I'm doing is I'm defining and say, this is what I want my graph to look like. In the additional instructions, that's where my prompt is. It's where I've added everything in there to do that. So everything is listed within this. I just analyze that, it just lets it do its stuff. When this read in, that's what it was using. If I look at my, let's take one of the, stay on. Hopefully this will come back quicker. If I look here, so if I ignore this, firstly, you're going to take out the document and chuck nodes, because those two are nodes which are just built by the graph builder itself. They relate to the actual document that was read in. What I've read in here is the Novo Nordisk pipeline. And I've only got the company, the disease, the molecules, and the targets, right? This isn't showing everything that's in there, but my model is now much easier. It's much clearer. I have in pipeline, I have targets, and I have treats. So I don't have all of that extra gubbins of the 106 different types of labels. I've just got the 4 I wanted. 5 I probably won't. Which means my queries can be more accurate. And this gives me, I still have the option of running my LLM questions, what molecules, drugs are being researched into what cancer, and I can do that and I can bring all this stuff in here. And that's fine. Like, whether you like a chatbot as a user interface for doing stuff versus you prefer to look at dashboards and stuff, that's entirely up to you. What I have done is generate a dashboard using another product we have called NeoDash Commercial. I assume commercial is part of the title, but it's NeoDash is the thing we're actually using. And I've taken that same data set we were looking at. So this is exactly what we've just been looking at. And I basically created a dashboard to allow your, as a researcher to go in and start to investigate certain elements of this. Obviously, the chat is something which I would add into this. Unfortunately, I don't have the add-in for this particular one right now, but here I've got a list of all the documents I've read in. I can see when I read them in, so I can keep track of everything that's gone through. But what I also have is I can see, Abve has 80 molecules in its pipeline. Roche has 80 in its pipeline. Bristol Myers Squibb has 74. And I might want to investigate. I can maybe click on Roche and go into there. I can see what molecules it is creating, which ones they're currently targeting. So Zofluza. Yep, Zofluza. So let's put you up here. Associated. So Zofluza is targeting, I'm guessing, and I'm going to be looking very heavily towards Alex at this point, influenza-based targets. I can see, and that's largely because I can see it's treating influenza. So we've got seasonal influenza, A and B, so A and B. The nice thing is I can see they're all associated. I can start to track all this stuff, and I can go through basically what Roche is looking at. And this is all very much from the point of view of Roche, right? So I'm coming from the company side of things. Back into our overview, I can also see that actually of all the companies I've read in, which are there are like 12 molecules within all of this data set at the moment which target the CD20 target. I don't know what the CD20 is. I don't know if Alex knows off by hand what CD20 is. Maybe he does. He's a big expert with those sort of numbers and letters. But for me, I'm going to click on it. I'm going to go into here and let me just click on one of the molecules. So I can see here that the diseases associated with CD20 are the B-cell cancers, non-Hodgkin lymphoma. I can see that this drug, Epovricotaka, well, close enough, why not? I can see what that is treating. I can see the targets it's aiming for. So I can actually see that CD3 and CD20 are both targeted by this drug. I can see the companies which are also targeting CD20. So Roche here has these molecules which are targeting it. Chugai, Biogen, like these are all the ones which are aiming towards targeting CD20. And I can see all the different diseases here, so even MS and things which are there. I can go back to my overview. Maybe I'm also interested in a particular type of disease. So I think Alex, from his experience, let's see if we have type 2 diabetes. So I can see I've got this type 2 diabetes here. I'm going to struggle to find that in here because of the names, but I'm just going to pick one of these ones. So I'm going to say retinal health, because that's, here we go. So type 2 diabetes, let's pick you. I don't know any members of that. Yeah. We'll go with solid tumors. So from a particular disease information, what I'm able to do now is I'm able to see that, okay, if I wanted to see which companies, so I want to start with solid tumors, I want to know which companies, so these are the companies that are currently investigating solid tumors. I can see I've got my solid tumors there. I can see Johnson & Johnson is working on J&J 9401, which treats solid tumors. Pfizer has these ones here. Genus has there. AbbVie's clearly spending quite a bit on there, but also potentially that these ones have worked in collaboration with Calico. So I can see that you've got the Calico ones all linked in, and they're all going through there. And up here, Bristol Myers Squibb. So I can start to analyze, like, if I'm researching solid tumors, who else is also doing that? What are they looking at? I've got a list of the companies down the side. So we can go back to there. And largely, now I've got all the information in there, I can start to ask more special questions to my LLM if I want to. Tell me the clinical phase. I can get them listed by company, listed by what they're doing. Obviously, quite a lot of these don't have the clinical phase. And I think it's important to bear in mind that the data that's put in, there are a few things which it's worth bearing in mind. We firstly went to a very generic LLM, so it hasn't been trained, means it doesn't necessarily know everything it's looking for. We would go to BioBert, or we start using the actual bio LLMs to do this properly if you want to do, or train it on our own data. If we had a mass pile of pipeline documents, we could do this on. Anything else which is, to do this in production, we'd be looking at far more passing this through a set of actual like probably Python scripts. You'd have a pipeline that brings all this stuff through. You wouldn't use the LLM builder to do that, but what you can do is the LLM builder to get an understanding for how to build your graph, for how to manage it, and to see how you can interact, and use it as a starting pad, a launch pad to get yourself into the place where you want to be. I think, right, that is me. Don't think there's any other thing at the moment. What I'm going to do is I'm going to do a bit of a camera swivel to bring Alex in, if he wants to.
Yeah.
Let me stop sharing somehow. There we go. Boom. Right.
All right. Thank you, Charlotte. We'll do that here from Charlotte's computer. So hope it was interesting to you, and this just gave you a little, I hope it sparked some ideas. As Charlotte said, it is really good for getting started without any technical expertises. Please don't use the LLM builder with proprietary data or with confidential data, because we are using a public LLM. So the data you put in gets for retraining. But if you want to know more, if you want to put that into more production-ready pipeline, you would use our GraphRag package. Yep. Most people are using Python for that. I don't see any questions in the Q&A panel here. So it must be that Charlotte was very clear, sharp as a knife. We really enjoyed the session. Thank you so much to Russell and to Maria for the great presentation. Thank you, Charlotte, for preparing the demo, presenting the demo, and showing us what can be done without any I wouldn't say technical knowledge. It's rather like without a lot of
minimal technical
yeah,
knowledge.
with minimal technical understanding. So yeah, please come back, ask questions, connect us on LinkedIn, write us an email if you have any further questions. If you want to start projects, well, we're happy to take them. Hope that was a good use of your time. Hope you enjoyed the sessions, and we're looking forward to seeing you on a conference, on the next event. We have Graph Summit London coming up soon. There will be other Graph Summits or Graph events in your territory, so watch out on our website. And yeah,
Thank you.
with this, thank you very much, and speak soon.